Good afternoon, everyone. Welcome to this symposium. So this is a symposium which is concentrated on treatment of cosmic allergy and the formulas we use, and it's entitled Making Informed Choices for Nutritional Management of Infants with Cosmic Allergy. I will serve as the chair, and we have two excellent speakers, Lucy and Claire, and I'll present them more in detail later. And after the two talks, we have a live and Q&A session, and you can just start writing your questions down so we have enough questions and discussions. Here are my disclosures. So this symposium is a Nestlé Health Science Symposium. I am myself an associate professor at the University of Helsinki Children's Hospital. I work in pediatrics and pediatric allergy. My research interest is in probiotics, allergy prevention, microbiota, and I also have fruitful discussions with excellent collaborators in Europe and US. So I'll just go briefly through to our team. Cosmic allergy diagnostics is not an easy task, especially if we discuss non-IgE cosmic allergy. It's very difficult to set a strict and proper diagnosis. And this is an old study, but it is a very good study. And this study focused on whether supplementing feeding at the maternity hospital, if breastfeeding is not enough, with cow's milk, hydrolyzed formula, or donated pasteurized breast milk. And of these 6,200 babies, 10% of the parents contacted the researchers for suspicion of cosmic allergy. 247 of those had a positive elimination effect, symptoms subsided, and they went on to a challenge. And this graph presents the results of the oral food challenge. And what you can see here is that the light blue bars are those that were challenge positive, and the dark blue challenge negative. So about half are negative, half are positive. And I've highlighted here the gastro symptoms, the non-IgE symptoms. They're more often negative than positive. And here are some symptoms like colic, crying, changes in stool frequency, color, whatever. So this is a difficult task. And the prevalence in this study was by 1.9%, which is an average in many studies, older studies especially. And this is a Europreval study published some years ago. It's a cohort of 9 European countries, and 12,000 infants were followed, and if they had symptoms related to cosmic allergy, they were invited for a food challenge, a double-blind food challenge. And here are the prevalence results in this figure, where you can see that it varies from country to another, and especially the non-IgE mediated are very different in different countries. Some do not recognize them, and some have a lot of them. So diagnostics are needed. Good diagnostics are needed. And this slide is a ESPGHAN position paper, a recent paper from last year, and just want to raise a couple of things from here. Even if there are suspicion of cosmic allergy, breastfeeding should be encouraged. This is our job as pediatricians, nurses, dietitians. And the diagnostics should be based on an elimination with a result and then a new challenge. And also, according to new studies, the tolerance development starts very early. So some of the kids do not read very strict elimination diets, and some can tolerate small amounts and increasing amounts. So this is a third very important thing from this slide. I'll also briefly touch the overdiagnostics, which is a problem, and unnecessary elimination diets are harmful, and they can put burden on the family, and they can have long-term effects on the child and the family. But after correct diagnostics, we come to the very important thing, that treating the kids. And this is a Jackman paper from last year, listing the important aspects you should take into account when treating a child with cosmic allergy. And the yellow encircling is the one we are going to focus on today. And now I'll briefly introduce our excellent speakers. First, Lucy Jackman, who is a clinical lead dietitian on motility, GI, and allergy at Great Ormond Street in London. She is passionate about advancing clinical care through research and education, a very good motivating thing. And her title will be, "It's not just about removing the cow's milk protein." And after Lucy, Clare Mills will be giving her talk. She is a professor of food and molecular immunology at Surrey University in UK. She is involved in many EU and WHO projects relating to food allergens. And her research interest has been long-lasting, relating to protein structure, what makes a protein an allergen, and also the matrix effect. And her title is, "Are all protein hydrolysates the same?" So please, Lucy, I call upon you to the stage.
So thank you for that introduction. And as Mikael said, my name's Lucy, and I'm a clinical dietitian working in London. So I'm going to be talking today about, it's not just about the removal of cow's milk protein. These are my conflicts of interest, and this symposium is sponsored by Nestlé Health Science. So I know that all clinicians in this room are well aware of what cow's milk protein allergy is, but just a brief update. So it's an immune-triggered reaction caused by whey or the casein, so the protein component that we get in cow's milk. And broadly speaking, we've got sort of three main types that we come across in clinical practice. We've got our IgE-mediated, non-IgE-mediated, and then those infants who also have a combination of IgE and non-IgE. With the non-IgE allergies in particular, the diagnosis can be quite tricky because many of the symptoms that we see are also sort of very common infant symptoms that you see in the majority of children. And so this can mean there can be a delay in diagnosis or a wrong diagnosis made. And we can see here in this study by the team from the EAT study is that actually, if we were using the IMAP guidelines, 3 in 4 children would be classified as a mild to moderate delayed cow's milk protein allergy. And so the true prevalence remains quite controversial. In the literature, it's reported around 1% of incidences of cow's milk allergy, but actually parental reporting is much higher, at around 10%. And the only way that we can diagnose these delayed allergies and delayed cow's milk allergy is using a process of the 4 R's. So we'd remove that protein, replace it with something suitable, reintroduce it after a set period of time, and see if there's a relapse in symptoms. And you might need to do this a couple of times to confirm that diagnosis. But the overdiagnosis occurs when this process of the 4 R's isn't completed. And unfortunately, this happens quite a lot in primary care. But even if we do suspect cow's milk allergy for any of our patients, we should recommend breastfeeding or breast milk as the optimal way of feeding these infants. And that's recommended in all guidelines as the first milk choice for infants. It's recommended from birth until 6 months of age exclusively, and then alongside complementary feeding until 2 years of age. And that's not just for the nutritional benefits. There's a number of other benefits that we see associated with breastfeeding. We've got our health benefits for the infant, but also for the mother. We've got social and bonding benefits between the infant and the mother. We've got economic benefits, so breast milk is free, doesn't cost the mum any money in formula or the healthcare service. And then we've got environmental benefits as well. So again, we're not using plastics, bottles, etc. if we're breastfeeding directly from the breast. From a nutritional perspective, breast milk contains pretty much everything that an infant needs. So this is a breakdown of mature term breast milk. This will differ from mother to mother, but broadly speaking, this is the average composition. And as you can see, this is predominantly made up of water, but we've also got carbohydrate, which is around 7%, predominantly from lactose, a fat component of around 3.4%, and then a protein component of about 1%, and that's casein and whey. We've also got these non-nutritional bioactive substances, so products like HMO, so human milk oligosaccharides, growth factors, cytokines, chemokines, and so on. And actually the only sort of vitamin and mineral that's not sufficient in breast milk is vitamin K and D, which is why we supplement that in our infants. But so in order to prevent overdiagnosis of cow's milk allergy or underdiagnosis, there's a number of guidelines. This is taken from the most recent ESPGHAN guidelines that was published last year. And as you can see, as Mikael's already shown, that breast milk is recommended as the first line. But actually, if that's not an option or the mother or family choose not to use breast milk, the next recommendation would be an extensively hydrolyzed formula, which would be either casein or whey hydrolysate or a rice-based formula. And then we've got our amino acid formulas that we use kind of as a last resort in these patients. So what do we have as options in terms of different formulas? So they're categorized into 5 main categories. We've got our partially hydrolyzed formulas, which are actually not suitable in cow's milk allergy. We've then got our extensively hydrolyzed formulas, amino acid formulas, soya formulas. In some countries, they're not available in the UK anymore, and then rice-based formulas. But actually, the decision about which formula we want to use should be based on the infant's symptoms, the nutritional composition that we need, and more recently thinking about whether there's any residual allergenicity of these hypoallergenic formulas. So when considering the management of cow's milk allergy using a hydrolyzed formula, it's important that we kind of understand the protein composition of cow's milk and the process that's involved in breaking it down to make it suitable for infants who do have allergies. So cow's milk contains 2 primary proteins. You've got your casein and your whey, and each of these are kind of broken up into a number of different allergic fractions, and they have different allergenicity depending on what type of fraction it is. Within these proteins, there's epitopes, and these can be conformational or linear, and it's in these epitopes where an allergic reaction is triggered. So for a formula to be extensively hydrolyzed, we need to break down the peptide bonds and the peptide lengths to alter the allergenicity to mean that they're not recognized by the epitopes and not able to trigger that allergic reaction. And broadly speaking, there's a sort of a 4-step process that's used in manufacturing these extensively hydrolyzed formulas. We've got heat treatment, which is then followed by enzymatic hydrolysis. some further hydrolysis using hydrostatic pressure, and then ultrafiltration of any of these residual long peptides. And this is the process that's used, by the majority of companies that produce extensively hydrolyzed formulas. But actually, when these were first brought out, processing techniques have moved on quite a lot. So it's possible that these formulas are maybe not as broken down or as hydrolyzed as we once thought that they were. And so that leads us to ask the question, are all extensively hydrolyzed formulas the same? And so they're all labeled as an extensively hydrolyzed formula, and they have a very similar nutritional profile, and that's tightly governed, and there's lots of regulations around what should and shouldn't be in infant formulas. But their composition can vary in terms of other components. So they might vary in terms of the type of protein in these formulas, so whether they're whey or casein. They might vary in terms of any residual proteins or peptide sizes, what the carbohydrate source is, so lactose or lactose-free, what the fat source is, so if it's MCT or an LCT-based feed, if there's any additions of things like probiotics, such as LGG, and then prebiotics as well, so HMO, GOS, and FOS. And so one of the major differences we see between these extensively hydrolyzed formula is the peptide length, or the Dalton size. And so the Dalton size is the molecular weight of a protein and the peptide. And at the moment, there's not a consensus definition of what an EHF should be. So the American Academy of Pediatrics recommend an EHF formula is where the most of the nitrogen is in the form of free amino acids and with the peptides less than 1.5 kilodaltons. BSACI, which is the British Allergy Society, recommend that they're products where the greatest percentage of peptides under 1 kilodalton. And then EACI suggests that these products should be tolerated by at least 90% of infants with confirmed cow's milk allergy. But either way, for us as clinicians, the peptide length and the Dalton size is not that readily available information. But it might be something that's really helpful for us to make a clinical decision about which formula to use if an infant needs an extensively hydrolyzed formula. And so I'm sure we can all think of those infants who we move on to an extensively hydrolyzed formula and their symptoms still persist. And this is similar to what we saw in the Europreval study, where they showed that less than 50% of infants with cow's milk allergy actually achieved a symptom resolution on an EHF. And so we need to further understand if this has an impact clinically. And so this was a study that was conducted by Nestlé Health Science, and they looked at all the EHFs that were available on the market at the time, and then analyzed them to look if there were what a percentage of the peptide lengths was still greater than 1,200 daltons. And as you can see, that there was quite a large majority, predominantly the whey-based formulas, that had a peptide length with a higher percentage of the 1,200 daltons. And so it's possible that infants could still react to these formulas. Oops. They also looked at whether there was any residual beta-lactoglobulin, so a type of sort of milk protein. And you can see that the products that had a higher amount of peptides with a higher length of greater than the 1,200 daltons actually had a higher amount of beta-globulin left in the product. The other factor that's quite variable in extensively hydrolyzed formula is whether there's any lactose added or whether they're lactose-free. And so if we think back to the composition of breast milk, around 7% of the composition is from carbohydrate, which is predominantly lactose. And this is a naturally occurring prebiotic, so that means that it feeds the bacteria that lives within the gut. And this is important because we know that the allergic infant has a slightly different microbiome compared to the non-allergic. And so they have lower numbers of bifidobacteria and lactobacilli. And so in this study, what they were looking at is whether the impact of adding lactose to an extensively hydrolyzed formula impacted the microbiome and also the metabolome. So that's what's produced by the bacteria within the gut. And so they found that the lactose could modify the gut microbiota in the allergic infants. It actually increased the total amount of bifidobacteria and lactobacilli, and it was associated with an increased concentration of short-chain fatty acids, which we know has a beneficial impact on the immune response, improving the epithelial barrier, and supporting sort of long-term health. Another benefit of having a lactose present in an infant formula is that it appears to increase the absorption of calcium. And so this study looked at 2 formulas, one with lactose added and one that was lactose-free. And they were comparing the amount of calcium and zinc that was absorbed in infants taking these formulas. And what they found is that the lactose increased the absorption of calcium but didn't have an impact on the zinc absorption. But it is important to say that there was still adequate calcium to meet the requirements for that infant in the product that was lactose-free. Here I just wanted to point out that actually there has been an update in the ESPGHAN guidelines around using formulas with lactose. And so historically, people have been a little bit wary about using an extensively hydrolyzed formula with lactose added when making the diagnosis of cow's milk allergy. But ESPGHAN suggests that there's not enough evidence that we should be avoiding lactose in cow's milk allergy anymore. And this should only be in an exception that if an infant has sort of severe diarrhea that's lasting longer than a week, which could be suspected as a transient lactase deficiency. And we might use an EHF without lactose in during the diagnostic process, but then with the view to transition back to an EHF containing lactose. As infant formulas evolve and science progressed, we're seeing more and more things added to the formula to make them as close to breast milk as possible. And one of the things that we're seeing added more and more is HMOs, so human milk oligosaccharides. And so this is a prebiotic that's naturally occurring in breast milk. So as I said before, this is a substrate that's selectively utilized by the host microorganism, which confers a health benefit. And so these HMOs are resistant to the pancreatic brush border enzymes and the low stomach pH. And so they're really selectable by the bacteria to produce lots of bifidobacteria and lactobacilli. There's over 200 structurally different HMOs that have been identified, and there are a number of them that are now being produced to be added to infant formula. And this is especially important in the first 1,000 days of life, so that's from conception, right through to age 2. Because we know that the infant's microbiome is continuing to develop until around 3 years of age. In a breastfed infant, the microbiome is predominantly populated by bifidobacteria, and HMO is highly selectable by bifidobacteria, which can ferment and produce lots of short-chain fatty acids, so in particular, butyrate. And so one of the drivers of lots of formula companies is to add HMO with the aim to modulate the gut to make it more similar to that of a breastfed infant. And so this study compared the microbiome and the metabolome of the infants who had an EHF supplemented with different types of HMO and one without. And what they demonstrated is that the addition of the HMO to these extensively hydrolyzed formula promoted a better enrichment of bifidobacteria and slowed the maturation of the microbiota. And actually, there was a stronger correlation if the supplementation was started before 3 months of age. And so this suggests perhaps this prolongs the window for early immune modulation and slows down the development of the microbiome to be more that of an adult composition. For us as clinicians, we want to know, is it safe to use these formulas that are supplemented with HMO, and are they tolerated by the children that we want to use them in? And so in this study, they were looking at tolerability and the safety of using the formulas that were supplemented with 2 different types of HMO. And they demonstrated they were tolerated by nearly 100% of infants in the study, and actually that they were safe for use in children with cow's milk allergy. For me as a dietitian, I'm obviously very keen to make sure that the children are growing well when they're having these formulas with additional HMO. And there's a number of studies looking at growth in a various host of different formulas with these additional prebiotics. But this was based on a whey-based lactose-containing extensively hydrolyzed formula that was supplemented with 2 different types of HMO. And as you can see, the growth in terms of length, in terms of their head circumference and weight, and then their BMI for age Z-score were all comparable to that of the control formula. And then so just to sum up, breast milk remains the recommended mode of feeding for all infants with cow's milk allergy, and we should be supporting families to continue breastfeeding if they want to. An EHF of cow's milk, so a casein-based or a whey-based, or alternatively a rice-based hydrolysate, should be used. And the addition of HMOs may be beneficial to support the microbiome to be more similar to that of the breastfed infant, and it also supports adequate growth. Thank you.
Thank you so much, Lucy, for excellent overview over the field. We move to the next presentation with Clare, and we take questions after Clare's session. And so please write down your questions for Lucy so you remember them. Thank you. So Clare, please.
Great. We're there. Brilliant. Thank you very much for the introduction. So I'm going to talk to you about protein hydrolysates and whether they are all the same. And I'm going to present some of the results of a project that I've been doing at the University of Surrey. So you can see up there my team that are listed, together with the team in Nestlé who supported it, and in Waters Corporation, a mass spectrometry company who actually were involved as well. And just to see my declaration of interest, so research was funded by Nestlé. I have a lot of other funding from the UK Food Standards Agency, the European Food Safety Authority, and the European Union. And I have a number of industrial collaborators. And the other thing is that I have founder shares in a company called Reactor Healthcare that produces foods for diagnosis of food allergies. So we've already had a great introduction from Lucy, so I'm going to probably repeat a bit of what she's actually already introduced to you. But the first thing I thought was looking at, I'm going to be focusing on IgE-mediated food allergies. And so what are the relevant allergens that cause clinical reactions in relation to IgE are caseins. And this is data from a wonderful systematic review of diagnosis of food allergy done by Carmen Reggioni, who's now in Toronto. So caseins, 93% specificity with a 1.8 kilounit cutoff, alpha-lactalbumin, beta-lactoglobulin. So basically, most of the proteins in cow's milk are clinically relevant allergens to consider. So what I got now is a structure. This is what cow's milk casein looks like. And actually, human casein is not dissimilar. So we've got about 80% milk protein, and it's got a flexible structure. So caseins are unusual. They're kind of quite wobbly and moving around a lot. What's also very special about them is they are what we call phosphorylated, so they have phosphate on them. And this is a really important property of caseins that enables them to bind calcium at concentrations way above what you can get to dissolve in just water. They are really special molecules. We know that these calcium binding of the casein form little nanoclusters, which have a size of about 6 nanometers, so that's millionths of a meter, so they're very, very small. And these aggregate to form what we call the casein micelle that you can see here, which is much bigger. And actually, these casein micelles are part of why milk looks white, because they scatter light. So they're actually really big. So that's a little overview of casein. And then if we look at our whey proteins, they're completely different. So these are what we would call globular proteins. So they don't have a wavy, wobbly structure. They're quite compact, and they're actually quite rigid, and particularly beta-lactoglobulin, which is a major allergen. We call it BOS-D5, about 2% of milk protein. It has a structural similarity to some of the molecules that cause inhalant allergens from inhalant allergens from animal dander. And it's very resistant to digestion, and it's also very resistant to heating. So sometimes when you heat proteins up, their structure changes. That's why egg white goes from being translucent to opaque when you cook it. The proteins all unfold, but beta-lactoglobulin is a tough beastie, and it doesn't unfold when you heat it up very well. So the other whey protein is this special little protein called alpha-lactalbumin, or BOS-D4. And this actually is very interesting. It has almost the identical structure to a protein you find in egg white called lysozyme. And we also make it in our tears. It helps have antibacterial effects in the eye. And it also binds calcium. But unlike beta-lactoglobulin, it's much less stable, and it breaks down when you digest it with proteases, and it unfolds when you heat it up. So those are our main characters, if you like, in the play today about hydrolysis of EHF. So those are the ones we need to think about. And the next thing, which Lucy also touched on, was this idea of epitopes. So the epitopes are basically the structures on a protein that an antibody recognizes. And obviously, when we're talking about IgE-mediated food allergy, we're talking about IgE epitopes, but the same rules apply whether it's an IgG or an IgM. So we have two different sorts. So the first one here is what we call a linear epitope. They're continuous, and they are comprised of a linear stretch of amino acids, just that straight amino acid sequence. And they're often quite flexible and found on the surface of proteins. But they're not the major type of epitope that we have on proteins. Most of them fall into this category of being conformational or discontinuous, where the protein structure brings together different elements of them to form the thing that the IgE recognizes. Now, when you heat up a protein, when you cook it and it unfolds, you lose these types of conformational epitopes, but these linear epitopes tend to remain. And that's how food processing in particular can affect the allergenicity of foods. And then the other thing that Lucy already alluded to is that if you digest it or you break the amino acids that are joined together in the polypeptide chain, if you digest it, that also can destroy those epitopes, and it can destroy both linear and conformational epitopes. So these are the sorts of processes we use in food that could modify our IgE epitopes. So extremes of pH, and acid is often used. You see it in yogurt. That's why lactic acid bacteria make proteins precipitate to form yogurt. We also have heating, roasting, steaming, boiling, and hydrolysis. And that's the one that's really important here that we're going to be talking about. You can use acid, you can use enzymes, or sometimes fermentation can also cause that hydrolysis. So when you have your infant formula, or this hydrolyzed formula, actually what you have is the process whereby you start off with your starting material and you treat them usually with acid or addition of enzymes. And generally, I think in infant formula, they use enzymes. You can start off with cow's milk. This is what's at the top end here, but you can then fractionate that to make whey or casein, and it then goes into this process of heat treatment and spray drying with the addition of the enzymes up here to cause hydrolysis. And then generally speaking, what you also have is ultrafiltration through a 5 kilodalton filter. So we can get two different types of hydrolyzed product that's used in infant formula. Manufacture the EHF formula, you've got partially hydrolyzed protein. So this has got peptides that are supposed to be less than 5 kilodaltons. And then you have extensively hydrolyzed protein where you've got peptides of less than 3 kilodalton. And as I said, that usually goes through some type of filter. So partial hydrolysis, we know, can leave IgE epitopes intact. And this is work that was done many years ago using plasmin degradation of bovine and caprine beta-casein. And we have very large fragments left. And that did not alter the IgE binding here when you can see the degraded or the non-degraded protein. So if you don't chop it up enough, you won't lose the IgE binding. Another study actually has shown that the IgE binding capacity is reduced as the extent of hydrolysis increases, but it doesn't actually abolish it. And this study showed that the IgE binding that was left was due to residual intact protein or large fragments of protein that remained. So this is the work that Sophie Newton published some time ago, showing in a whole study of all sorts of different types of hydrolyzed infant formula that there was a huge heterogeneity in the level of hydrolysis, which you can see here in the bars, and then the yellow dots show the levels of residual BLG. So this is the same as Lucy actually showed. And as a consequence of this work, I actually were working with the Nestlé team. We put a project proposal together to actually map that composition in more detail, to characterize it regarding the residual intact protein, the peptides that were maybe 1,300 to 1,600 daltons. So that's a peptide that's large enough to have an IgE epitope, and whether they also formed aggregates. And those aggregates can be formed by cross-links we call disulfide bonds, or perhaps through interactions with other constituents like the calcium, which, if you remember, interacts with the casein in those nanoclusters. Now, we didn't get funding initially when we bid for it, so Nestlé decided to fund it directly. And some of the work has been done by the team in Nestlé who sourced all the formulas. My team have done the sample preparation and biochemical analysis and data analysis, and Waters Corporation have done the mass spectrometry analysis. And both my team and the Waters team have done this with blinded samples, so we have no idea what's what. And these are the sorts of methods we used, something called SDSPGHAN, which gives us profiles of proteins and peptides from 3,000 up to 200,000 daltons. That's very big. We looked at aggregates using another technique called gel permeation chromatography, where we use conditions that mimic those found in infant formula with a lot of calcium. We nearly broke the equipment doing that. And then we have this very posh mass spectrometry, which is really clever because it actually enables us to sequence the peptides so we could actually see what had happened to the IgE epitopes. So, step one, residual protein determined by SDSPGHAN. So this is the SDSPGHAN analysis. What you've got up at the top here are the very big proteins. Here are the very small ones, which is what we're aiming to have in our EHF. So you can see here, partially hydrolyzed formula, a whole spectrum, smeary spectrum. It always looks like this when it's been heated. No clear BLG in there or anything else, but a lot of big stuff, even going right up to here, which is over 100,000. Very big things. Definitely would cause IgE reactivity. So then in our EHF, what was really surprising, EHF1 has a lot of stuff in it. It's much less hydrolyzed. And what we saw was EHF2, they still got a bit, EHF3 has, and then we get to 4 and 5 and 6 and 7 and 8, and they really don't have anything in. So this very much confirmed what Sophie had observed in her study some years ago. Not all EHF are the same, and some of them really have quite significant amounts of intact or aggregated protein. So the team in Nestlé repeated the same analysis that they did before, Sophie Newton and Mikael Affolter, and saw exactly the same sort of thing. Here's our PHF. Never give that to a child with cow's milk protein allergy. And this blue bar is showing the amount of peptides, more than 1,200 Daltons in size. The red dot is the residual BLG, the green dot is the residual casein. And what you can see is EHF1 and 2, and actually 3 as well, have a lot of stuff still left in them by these, determined by these other methods. But when we get to these other EHF, there is virtually nothing in there. So then we go on to the mass spec. Now this is really complicated. This is for those who are really interested. It's a principal components analysis. But the thing that I want to show you here is when we did the mass spectrometry analysis, EHF1 and the partially hydrolyzed formula clustered together down here. EHF2 and 3 went up to the top, and then all the others sat together here. And those differences reflect the number of peptides, the size of the peptides, which in turn reflect things like the starting materials that someone might have used in their EHF, the enzymes, and other types of processing parameter. So we've also managed to look at the peptide size distribution and the number of peptides that we see. So with a mass spectrometry, we could see that in our PHF, we don't have that many big peptides. That's probably because the protein is not very well hydrolyzed and our mass spectrometer can't see it, but that had a lot of peptides. So did EHF1 and EHF2, and you can see it's gradually going down. So we get to EHF8 that only had 74 beta-lactoglobulin peptides that we could sequence. That formula also had no peptides over 3 kilodaltons in size, whereas actually some of the others, and you look at EHF1, they have quite a few big peptides. So I said to you, this technique that we've used for the mass spectrometry gives us the sequence of those peptides. And this is an example of one of the longer peptides that we found in beta-lactoglobulin. And the important thing is the levels of this varied in the different EHF. This is the sequence of the peptide that we got. It's got a molecular weight of 4,000 Daltons, so that's quite big. But it would have gone through that 5 kilodalton filter if the manufacturers had been using it. And what you see here highlighted in the kind of pale orange, that is the site of one of the IgE epitopes that have been identified by a couple of different teams in beta-lactoglobulin. So what you can see here, we couldn't see this peptide in the partially hydrolyzed formula. That may be because the enzyme that was used didn't produce it or wasn't degraded enough. But we could see significant levels, and these are significant levels, in many of the infant formula until we get to EHF7, where there were only tiny amounts, and EHF8 had none of it. So you can see there, we've used a whole battery of techniques to really show that a lot of EHF that are labeled as EHF are not extensively hydrolyzed. They are not all the same. I think the physicochemical analysis we've done in this study explains some of the clinical observations, and there's a couple of citations down here at the end that say that EHF can cause symptoms, and highlights that not all products, not all EHF are the same, and that we actually had one that looked a bit more like a PHF. What we want to do next is, I've done biochemical analysis, but it's always important to link that to biological activity and whether we can look at effector cells, so basophil activation tests, for example, using biological samples from infants with a diagnosed cow's milk allergy. And that will help to validate the physicochemical analysis we've done. And what we hope that it will do is pave the way to better criteria. And Lucy had a really nice summary of those different criteria that there are out there. And if we can get a bit more science into that and make it a bit tighter, we can make it clearer what does it mean to have an EHF, and that might help support clinicians in choosing the right one for the right baby. And I think what we would hope in future is that we can have some kind of multi-stakeholder discussions about this with everybody, from the patients through the manufacturers, our medical societies, basic scientists, regulators, and obviously our all-important clinical and health professional teams caring for it. So, thank you very much for listening.
Thank you, Claire, for a nice presentation, a lot of information, a lot of data. So now the floor is open for discussions. Please go to one of the microphones in the rear or front in the corridors, and just raise your hand so I can see you. Any question is a good question. Please.
Thank you so much for these elegant presentations. My question about Dalton size, is it important only for IgE-mediated allergy, or it having an importance for non-IgE-mediated allergy?
Well, we know that the hydrolysis, for example, that we've been focusing on is to remove the IgE epitopes. However, one of the outcomes is if you have bigger peptides, say 9 residues, that you have the opportunity there to retain the T cell epitopes. Now, I don't know to what extent, and I'm going to look at Lucy's going to get horrified because I look at her and say, do you know the answer to this? But I think retaining the T cell epitopes provides the opportunity for the immune system to still be activated. And I know more about celiac disease than I do things like eosinophilic esophagitis, and there, 9 residues is enough to actually get that T cell activation going in celiac disease. So there are principles there that you would keep some of that activity, but I don't know that it's actually ever been shown in patients with EoE. So over to you, Lucy.
Yeah, so I think, there, in EoE in particular, there is some work going on around whether extensively hydrolyzed formulas can be used in that patient group, but the results have not been published yet. So I think from a non-IgE mediated perspective, there is potential that these residual proteins could still trigger that allergic reaction, for an infant with a non-IgE, but also for other children who might have things like eosinophilic esophagitis as well.
Okay, thank you.
Please, in the back. It's not on?
Oh, it is.
The mic is not working in the rear. Oh. No problem.
Hello. Right now, if I understand right, this number 7, number 8 are probably more hydrolyzed formula than compared to the other. So does it affect the taste or the acceptance by the baby? Have you looked at that? Because obviously the difference from amino acid is the taste and acceptance of the children.
So I don't think that's been looked at just yet, following on from this study. we obviously, these studies or these products are blinded, so we don't know which product they are. I know there has been lots of tolerability and taste acceptability studies done on all of the formulas. So it's possible that that data is already out there, but I suppose we'd have to unblind the samples to know if there was a correlation between better tasting or
That's
not.
the balance we have to get between hydrolyzed formula and the acceptance.
Yeah.
That is also the...
But I suppose, although it wasn't presented today, we know that the lactose in a EHF often also increases palatability, makes the product slightly sweeter. And so infants often do accept it better. So I suppose that's another reason why we might not want to just blanket, not give a formula with lactose in it as well.
Thank you so much. So can I say 7th and 8th from Nestlé? Or is it not known? It's not disclosed.
So, well, it's your study, but the study was blinded. So, yeah, we don't know.
I don't know.
Now, say it, please.
Yes. Lucy, in Latin America, we are only 500 years in taking cow milk. In Europe, many, many thousands of years. What's the reason that the frequency of allergic is the same in Latin America and in Europe if we don't have the heritage environmental conditions?
Sorry, can you ask that again? Sorry.
Yes?
Yeah.
In Latin America, the babies, we are only intaking cow milks by 500 years ago.
Okay.
Before the conquest, we don't have cows in America.
Okay.
What's the reason? Is the same frequency in Europe and in Latin America? Because in Europe, you are intaking cow milk for many, many hundred years ago.
I mean, I probably don't know the answer to that question, if I'm honest. I mean, we know that the prevalence of allergy is increasing worldwide. And I suppose there'd be a number of different factors around that. Probably it might be sort of the development of the microbiome, the environmental changes, and all of those things which have an impact on sort of the development of allergy. So that would be my thoughts, but I don't know if you two have any other.
Perhaps if I can also say, because in the Europreval study, we did see exactly that heterogeneity. And in the UK and in the Netherlands, we had a much higher incidence of cow's milk protein allergy, but in Greece, it was really low. In fact, the prevalence of food allergy in infants, school-aged children and adults was much lower in Greece. So it does point to the fact that there are big environmental factors, and same happened in India, south of India, almost no food allergy. So there are changes going on in our environment and our lifestyle, I think, that do impact on that allergy rate. And to be honest, I don't think we found any answers so far.
Yeah. Thank you very much.
I might add that the awareness of cosmic allergy differs between countries and continents as well, among the parents, among the healthcare workers. So that might also be one player.
Thank you.
Just a question for
Yes.
Lucy. You had mentioned that we can try a soya product before we try a hydrolyzed milk product because of the cost factor involved. It's cheaper. So do you take a trial with a soya milk product in a newborn before you try a hydrolyzed product and then you go on to an amino acid based product? Or do you directly go to a hydrolyzed product when you're suspecting cow milk protein allergy?
So we would, as the majority of the guidelines are, we'd go to an extensively hydrolyzed product, so either a cow's milk hydrolysate or a rice-based formula, rather than the soya formula. Actually, in the UK, we don't have access to soya formulas anymore, so we wouldn't use those anyway.
Because the cost of a soya formula in India is 1/3 the cost of a hydrolyzed or an extensively hydrolyzed, and 1/4 or 1/5 of the price of an amino acid based formula. And there is no funding from the government for the parent, for the child, right? So would you advise us to try a soya formula if it's available in our country, or would you advise us to go for an extensively hydrolyzed formula?
I mean, I think the evidence is more for the extensively hydrolyzed formulas at the moment. So that would be the first line, and that is the first line in the guidelines. I appreciate the cost is a lot for these products, but I suppose that's where we should really be investing and encouraging breastfeeding, supporting these mothers to breastfeed rather than moving over to a formula more quickly than we should be, perhaps.
All right. But there's nothing legally wrong in trying a soya formula, right? In newborn baby.
There's no legal legal things as far as I'm aware. I'm not a lawyer. Okay. But we don't use soya-based formulas often in under 6 months.
All right.
Because of the phytoestrogens. So that's one of the reasons we wouldn't use it early on.
Thank you.
One more question, please.
Hi, thank you. This is a question for Lucy. I know you see a lot of children with EOE, and I'm wondering what formula you use for those.
So if we're using a formula, which the majority wouldn't require a formula, it would be an elemental, so an amino acid-based formula as a first-line choice in eosinophilic esophagitis. That would probably be the only caveat where we would use an amino acid formula as a first.
And based on your data, would you not consider some of the EHFs?
So at the moment, there's not much data. I suppose, we've seen that they're so different. If we choose one that's actually more like a partially hydrolyzed, we could trigger the eosinophilic esophagitis. So the data and the guidelines at the moment do recommend amino acid as first line if you're using a formula in EOE.
Okay, thank you.
So I might just conclude this session. Thank you very much to both speakers.
I have.
Sorry. Okay.
Thank you very much for the speakers. I would like to have an idea if I understood properly. Well, Dr. Mili, you said that sometimes the extensively hydrolyzed, the particles aggregate together, and so it causes a problem. Do you mean that you are going to have new benchmarks for more hydrolysate, less Dalton than this?
So that is exactly it, that there are some of them that have big aggregates in them, and they will not behave the same way when you give them, and the sorts of reactions you are going to get are going to be probably much more reactive than a formula where there are none of those aggregates there and the peptides are really much smaller. And we do need better, I think we need better specifications of what an EHF actually is.
So you are expecting that we go to what level of the Dalton?
So I think one of the things we need to do, because these data are really hot off the press and some of them only came about 3 weeks ago, and I think we need to go through them more carefully to see what actually would be perhaps the boundaries that you could have. The other thing that I think will be very important is that clinical reactivity, which I know the Nestlé team are very interested, so that you can connect the size and the aggregation directly to the sorts of reaction that Lucy was talking about there and what might be suitable for someone with an IgE-mediated allergy or someone with EOE.
Yeah. Thank you very much.
Okay, so I will close the session. Thanks you very much to both Lucy and Claire. Very clear and nice presentation. Thanks to the audience for being here, and thanks for all the good questions. And please give us feedback so we can develop these symposiums. Just scan that and give us our feedback. Have a nice Congress.